This video with slides aiming to explain how air pollutants cause lung cancer. Hi, I'm KM, an associate professor from the School of Life Sciences, the Chinese University of Hong Kong. Let's start with a chemical called benzoate-pyrene, which, after metabolism, is converted to DNA adduct of pyrene diol epoxide to bind DNA at the center of a double helix of DNA structure. Normal detoxification steps of the chemical benzoate-pyrene, representing a group of polyaromatic hydrocarbon chemicals, are shown in this slide. P450 enzymes could oxidize it, forming different epoxides or oxides. Following the action of epoxide hydrolase to remove the epoxides, further actions of phase 2 enzymes to remove these toxic chemicals from our body. However, these epoxides are highly reactive and may become resistant to epoxide hydrolase. With high concentrations, they can attack DNA, causing mutations. If the mutations are with the oncogenes, such as the RAS oncogene, lung cancer and skin tumors are resulted. This slide shows the structure of the pH polyaromatic hydrocarbons, anti-diol epoxides found as metabolites in our body affected by benzoate-pyrene or lung cancer patients. As a result, the P53 mutation hotspots include several codons, also found in lung cancer patients. DNA damages are found. Suppose we have our own PARP repair mechanisms of poly-ADP ribose polymerase for incision of DNA-based lesions. But as the concentration of the chemicals could be high, leading to P53 and P21 gene mutations, causing apoptosis and cell cycle arrest. P53 is a tumor suppressor controlling normal cell cycle to occur, but after DNA damages, P53 mutated could have negative control of cell cycle and apoptosis. They act on DNA directly. Other biochemical responses to DNA damages also include the P21 RAS or RAS oncoprotein. It controls cell cycle, and if it has mutation, cell cycle could not be controlled. Uncontrolled cell cycle leading to cell growth and tumor formation. This slide shows the intrinsic and extrinsic pathways of apoptosis, programmed cell death. BCL2 is such factor could block the extrinsic pathway. In lung cancer cells, with the P53 DAP kinase pathway, methylation of genes and BCL2 levels both increase to block apoptosis. While the P53 BCL2 induced in non-small cell lung cancer showing a low apoptotic index, SCLC small cell lung cancer has a different phenomenon of high apoptotic index. These two types of lung cancer cells have different outcomes. Finally, cell cycle is controlled by growth factors. To turn on cell cycles, RAS oncoprotein is such factor mediating the cell cycle effects of growth factors on target genes for cell division. This RAS protein is also called P21 RAS or just P21. It promotes cell growth, and when mutated, mutations of the proto-oncogenes would cause lung cancer and uncontrolled cell growth. P21 mutations found in many non-small cell lung cancer, forming adenocarcinoma in the lung. The last agent or group of chemicals is heavy metal ions, For example, chromium-6 and cadmium. They are cancer promoters as they generate reactive oxygen species, ROS, initially cause apoptosis in some cells and eventually promoting p53 damages, promoting lung cancers. To sum up, how air pollutants cause lung cancer. Metal ions can induce ROS, reactive oxygen species, inducing cell death of lung epithelial cells. pH cause oncogene mutations like p53 and p21. Many diesel exhausts also induce the mutation frequencies, potentiating the effects of the pathogenesis of lung cancer. In these slides and videos, we have reviewed how air pollution are related to higher hospital admission and death rate, how air pollution can cause lung cancer. How about indoor air pollution? What chemicals could also do harm us? Explain why they are toxic. You may like to do revision papers on these topics to reinforce your understanding of how air pollution kills. Goodbye.